Hello, everyone. My name is Denizard Bueno. I'm a PhD candidate at the Federal University of Viçosa in Brazil. I'm currently working as a visiting scholar here at the University of Illinois under the supervision of Professor Alex Lipka. My advisor in Brazil is Professor Deli Silva. And today I would like to talk about applying generation mean analysis to tomato plant architecture breeding and how I'm integrating spatial modeling and environmental covariates to improve genetic inference. So, ideal plant architecture has become increasingly important in modern agriculture. Over the past decade, there's been a growing demand for cultivars that are compatible with mechanization, suited for greenhouse and urban farming, and capable of delivering higher fruit quality. These new production systems require cultivars to be specifically made to grow in this kind of habits, and they have to be stress resistant and disease resistant. So in our study, we aim to assess whether incorporation spatial data and irrigation-related environmental covariates could enhance our ability to capture the genetic architecture underlying these traits. So we use a biparental cross between a commercial tomato variety, here designed 40 from our accession, and a dwarf genotype designated here C4, which has been proved to be useful for industry usage. It's a dwarf variety. It comes from the vegetable germplasm bank in Viçosa, and it's critical for plant architecture breeding because its genes uh, completely alter the shape of the traits, and the cultivar 40 is specifically selected for disease resistance. Uh, the cross we produce 6 generations of these biparental crosses. These were planted in a completely randomized design inside the greenhouse. The number of individuals per generation was proportional to the expected segregation ratios. For example, the F2 had more individuals than either the back cross populations. Uh, and here the pedigree tree shows how the crosses were made to obtain the populations in this study. So here is a picture of the growing season. Plants were evaluated after 80 days of transplanting. The phenotyping was done in multiple traits. Here is the traits that were evaluated in this study for plant architecture. So our analysis follow a two-step approach. First, we use generalized linear mixed models, incorporating spatial data and environmental covariates. This heat map shows the spatial distribution of irrigation uniformity across the greenhouse. We include irrigation as a covariate, and the residual structure used in this model was the autoregressive uh, correlation, which accounts for less correlation of variances on individuals that are further apart. So we compared several models to determine the best fit, and across all traits and these models, the irrigation covariate was not significant, statistically significant, and therefore it was removed. In contrast, the autoregressive correlation residual structure was very efficient throughout all the traits. This figure demonstrates how the applying this approach could reduce the environmental variance and increase the genetic variances. That is what we expect from this study. Here are the variograms obtained by the autoregressive structure correlation system. So what we expect here is increase of variance across distance. So the x and y axis display the distance from individuals related to each other, and the z-axis show the semi-variance of the traits. So here we have like most of the traits fitting the expected results. Uh, and now we followed for the second stage, where we conduct the actual generation mean analysis. For this, we use predicted data and the original data set, and we extracted means and variances for each generation using these formulas, as suggested for Matter and Jinx. And first, we fitted the additive by dominance model and performed a goodness of fitness test to check if it was accepted. And here is the obtained table for height to the last leaf, or plant height. And here we already have values displaying that the expectancy of means for the additive by dominance models was reduced, which means the relative contribution for the means were diminished, which was what we expected to happen, because we expect a higher signaling from the genetic architecture using the predicted model when we model better the residuals. So here, the goodness of fit for the additive bidominance model was 91.79%. It was a subtle difference, but nonetheless was in the direction of improvement of what we expected to fit. For when we rejected the additive bidominance model, when the fitting was less than 85%, we fitted the epistatic model with the digenic interactions A by D, and D by D. And here we also see the same trend where the relative contributions for the mean is diminished. There's a reduce on the p-value or the significance of the parameters. So overall, we had an improvement on the estimates for the generation mean. As final remarks, I would like to mention that spatial modeling led to small but consistent improvements in model fitting and inference quality overall. This enhancement are specifically important in real-world breeding situations where we have to handle large populations and evaluate them under non-ideal and uncontrolled environments. Also, GMA proved to be a flexible, insightful tool to dissect the genetic architecture. We were able to fit general models for most of the traits, and future work could expand this to additional traits or more complex, like multi-trait models. We plan to export this multi-trait approach to account for genetic correlation among plant architecture. And although irrigation was not significant in our models, might due to uniform water diffusion in soil and the plant's ability to compensate dynamically. Nonetheless, it's important to point that the testing of environmental covariates in field trials. I would like also to thank Professor Alex Lipka, our lab mates here at the University of Illinois, for their support, as well Professor Delene Higgins and my colleagues at the University of Viçosa. This research was supported by FAPEMI, CAPES, and CNPq, which provided both scholarship and materials for this research.